Good afternoon. I'm Dr. Brian Shapiro, a scientific content specialist at ATCC. Thank you for joining us for the latest installment in the ATCC Excellence in Research webinar series entitled Best Practices for Cell Culture, presented by Mr. Stephen Budd. Mr. Budd is a product line business specialist at ATCC. In this presentation, Mr. Budd will tap into ATCC's vast cell culture experience and share the best practices for culturing cells that ensure optimal results and performance. He will cover all aspects of successful cell culture, including culture initiation, expansion, authentication, cryopreservation, and transfection. If you have any questions for our speaker, please use the chat function available through the webinar program. All questions will be answered as time allows at the end of the presentation. The recorded webinar presentation will be archived on the ATCC website, www.atcc.org. So with that, I'd like to welcome Steve Budd.
Thank you, Brian. Founded in 1925, ATCC is a nonprofit organization with headquarters in Manassas, Virginia, and an R&D and service center in Gaithersburg, Maryland. We're the world's largest, most diverse biological materials and information resource for cells and microbes, the gold standard, which leads us into the topic of today's webinar on cell culture fundamentals. In today's presentation, we're going to start with cell handling tips, focusing on cryopreservation, aseptic technique and contamination, cell authentication, and issues with mycoplasma. We'll then discuss reasons for choosing certain cell types from primary cells to cell lines, and finish with some points on transfection and cell viability. So we're first going to start with some cell and media handling techniques. So to start with thawing, when ready to thaw a vial of cells, the most important factor to note is that unlike freezing cells, thawing cells should be formed as quickly as possible to remove the cryoprotectant from the cells. For best results, place the vial in a 37°C bath for approximately 2 minutes, gently agitating the vial to break up the ice. After removal from the bath, make sure to spray the vial with ethanol for decontamination. Transfer the contents to a sterile centrifuge tube with about 9 mL of complete media, which includes a cell culture medium and about 10% of FBS. When adding the growth media, pipette the liquid into the tube slowly in a dropwise fashion to avoid osmotic shock before adding the final full amount. When done, centrifuge the tube for 10 minutes at 125 Gs, discarding the supernatant, and resuspend in 1 to 2 mL of growth media. Place the cells in the desired cell culture vessel with additional media. The main take-home here is to thaw cells as quickly as possible when placed in the water bath. Also note that for most primary cells, it is usually advisable not to centrifuge the cells, as this could be detrimental to the cell health. Simply plate the cells after adding the additional growth media. So after thawing cells, they should be plated in an appropriate vessel, which could be cell culture dishes or flasks. This is all depending on your specific needs. For most cell types, the general accepted practice is to allow them to settle for about 24 hours after seeding. At this point, the cells should be checked for confluency, which is the percent coverage of cells over the surface of the vessel. If expanding cells, the ideal time to cell culture is at approximately 80% confluency. The time it takes to reach this level can vary upon cell types. Primary cells can take several days or even up to a couple of weeks. The diagram gives a good visual of the different growth phases of cells in culture. When first plated, cells enter a lag phase where they grow slowly while trying to recover from the stress of subculturing. Cells then enter the exponential phase of growth. Here, cells will grow exponentially as long as growth surface is available. When this surface is completely occupied, cells then enter the stationary phase of no growth. If they persist in this phase, they will then enter the decline phase, not shown in this graph, but to the right of the stationary phase. At this phase, they will lose viability and essentially die. To ensure phenotypic stability, cell lines need to be maintained in subcultures in the exponential phase. This will need to occur before a cell monolayer becomes 100% confluent or reaches its maximum recommended cell density. As stated before, that ideal confluency is about 80%. So when you've reached 80% confluency, cells can be passaged with trypsin EDTA. Trypsin disrupts the proteins involved in attaching cells to the culture vessel. The figure on the right shows a progression of cells being trypsinized. At the starting point, we see cells attached as a monolayer. After trypsin is added to the cells, they become flattened and more round. When completely detached, they can be seen floating around in clusters. This should take about 3 to 5 minutes, but make sure to note that the dish should be gently agitated to help detach the cells, otherwise it could take longer. Long-term exposure of trypsin to cells can be detrimental, so the time in trypsin needs to be minimized. Also note that if using primary cells, a special lower concentration of formula of about 0.05% trypsin should be used. The lower concentration of trypsin will definitely require the physical agitation of the cell culture dish to ensure detachment. Also, when trypsinization is complete, the reaction needs to be quenched, ideally with a soybean neutralizing solution. Again, this will help protect the cells from any damage of long-term exposure to trypsin. In the next several slides, we want to discuss techniques on cryopreservation. I want to first go over the basic theory behind cryopreservation and how it works. So the freezing process is a complex phenomenon that's been studied for decades. Cellular metabolism stops when all water is turned to ice. This, however, is largely detrimental to the cell's health. Two main reasons for this: ice formation and an increase in solute concentration within the cell. This is caused by either freezing too quickly or too slowly. The solution to this problem is maintaining the ideal freezing rate while using a cryoprotectant, the most commonly used being DMSO. The combined effect will minimize ice crystallization and reduce the solute concentration effects. And the important take-home here, as the diagram notes, for most cells, the optimal rate of freezing is a drop of 1 to 2 degrees Celsius per minute. So freezing cells is typically a two-stage process in which cells are incubated for some time at minus 70 degrees Celsius before being stored at minus 140 degrees C. As previously discussed, cells must be cooled at a rate of about minus 1 degree Celsius per minute. This can be achieved with the use of a controlled rate freezer or controlled rate chambers. The length of time needed in the minus 70 would depend on the type of chamber used. So after spending the appropriate time at minus 70 degrees C, the vials must be transferred to minus 140 C for long-term storage. It's recommended to store cells in liquid nitrogen tanks for this purpose. So as I just stated, long-term storage of mammalian cells require that cells be exposed to temperatures below minus 140 degrees Celsius. At these low temperatures, metabolic activity ceases, allowing for the preservation of cells for an indefinite period of time. For storing biological material at these low temperatures, it is highly advisable to use liquid nitrogen freezers. So there are two basic types of liquid nitrogen storage practices. One is immersing the vials directly in the liquid. The other involves holding the vials in the vapor phase above the liquid. Vapor phase systems create a vertical temperature gradient within the container. At the very bottom, where the liquid nitrogen resides, the temperature will maintain about minus 196 degrees Celsius. The temperature in the vapor nitrogen decreases as it reaches the top portion of the container. Enough liquid nitrogen should be used to guarantee that the warmest part of the container at the top is always at a temperature at or under minus 140 degrees Celsius. It is highly recommended that vials be stored in the vapor phase rather than the liquid. Since metabolic activity is arrested at minus 140 degrees C, the additional cold temperatures at the liquid phase offers no real advantage. However, immersing the material in the liquid phase runs the risk that some of the liquid nitrogen could penetrate the vials and cause them to crack or even burst. As we leave the discussion on cryopreservation, I want to discuss cell characterization as it relates to our upcoming topic of general cell health, contamination, and authentication. It is necessary to characterize cells to determine how healthy and viable they are, as well as making sure that they're the cells you think they are. Simple cell viability counts using trypan blue with the hemocytometer are an easy way to determine the percentage of live cells over dead cells. With this method, live cells appear to have bright centers while dead cells appear dark blue as the dye penetrates the compromised membrane. Often when researchers receive cells from collaborators or other unreliable sources, they may find that the cells are not exactly what they thought they were. So morphology is a quick way to distinguish broad cell types from one another. In the example on the right, you have two cell types, fibroblasts and HUVECs. They have very distinct morphology. Fibroblasts tend to be elongated, whereas HUVECs tend to have a rounded cobblestone appearance. Also, since different cell types grow at different rates, always make sure your cells are growing the way you would expect. If cells are contaminated with another fast-growing cell, the growth rate and doubling time may be greatly increased. We now come to the topic of contamination. So the main types of contamination we'll be concerned about are microbial, which include bacterial, mycoplasma, fungi, and virus. There's also cellular contamination, meaning one cell type or cell line has invaded another. We'll discuss this in a little more detail shortly. So bacterial and fungal contamination are the most common problems seen in labs. They often appear quickly, but fortunately can be detected very easily. Turbid and acidic media usually indicate the presence of bacteria. Cell stress from microbial contamination may also lead to morphological changes in the cell type. Fungal contamination is seen by the appearance of obvious filamentous structures in the media. So at least one good thing about this microbial contamination is that it's very easy to detect quickly. Mycoplasma and cellular cross-contamination, discussed in just a minute, is not as easy to detect. So where does contamination come from? For starters, anything that has been contaminated in the past can contaminate cells going forward. Media and reagents that are not handled correctly can easily be a vehicle to introduce bacterial and fungus into cell cultures. Using items such as petri dishes or pipette tips that are not properly sterilized can certainly introduce contaminants. Personnel can also introduce particulates and aerosols on personal clothing or even on dry skin. Improperly maintained equipment can prevent protection from the entry of contaminants. Overpacking autoclaves or ovens can even cause problems with uneven heating, creating pockets of non-sterile supplies. The flow rate of hoods, if not properly adjusted, can allow particulates to enter. So what can you do to make sure cells and culture do not get contaminated? So I'm going to point out a few tips that should prevent contamination problems. First, in general, you want to make it hard for contaminants to enter your cell culture vessels. Small dishes, such as 35 to 60 millimeter dishes, can be placed in larger ones when in the incubator. Placing plates and dishes in larger ones are also ideal for transport. This adds an additional physical barrier that prevents particulates from entering. When using flasks, it is recommended to use vented caps that can be sealed tightly, but still allow gas exchange. I always use disposable aspirators and pipette tips that are pre-sterilized. Make sure that your hood has adequate laminar flow and never overcrowd or leave materials such as boxes or bottles in the hood indefinitely. This can impede proper flow and block attempts at cleaning and wiping down the hood surface. When bringing media and reagents into the hood, always spray the items down with alcohol to remove any contaminants. It is also advisable to bring in small amounts of reagents into the hood at any given time. If possible, you may want to aliquot small amounts in these containers to reduce the number of times you're bringing a possible contaminant into the hood. For instance, if a vial is contaminated, at the very least, it won't continue to enter the workspace. Always remember to wear clean lab coats that cover most of your arms to prevent dust and shedding skin to contaminate your cultures. We advise not using antibiotics in culture. There are a few reasons in addition to the potential of toxicity. If you use media with no antibiotics, and a contaminant enters your cell culture, chances are it will be visible within a couple of days. You can then discard the infected dishes. However, if there are antibiotics present, it may slow the growth of the contaminant, allowing further passages and freezing down your cell lines. In short, you may be saving large amounts of contaminated cells that can cause problems down the road. Resistant mycoplasma may also get introduced along with other bacteria. The antibiotics may get rid of the larger bacteria, but mycoplasma may persist undetected. So in short, antibiotics can also sort of cause this sort of sense of false security. When is it acceptable to use antibiotics? So a few exceptions make it necessary. For instance, if you're isolating primary cells from original tissue, it may be a good idea to use the antibiotics for the first couple of weeks. So mycoplasma contamination is a separate contamination issue that is extremely difficult to detect. So contamination from mycoplasma can cause a host of problems that includes altering the cell's function, growth, morphology, causing chromosomal aberrations, affect nucleic acid synthesis, and changes in membrane antigens and gene expression. The main source of mycoplasma contamination is receiving infected cultures from other research laboratories. Reagents purchased commercially can also be a source of contamination. Mycoplasma contamination can be detected through a number of available kits in the market. The Universal Mycoplasma Detection Kit from ATCC is a PCR-based kit that can detect 60 of the most commonly identified mycoplasma species. So to put it in perspective, over 90% of contamination is caused in just a few species. The Mycoplasma Detection Kit comes with specific primers that are unique to mycoplasma with mycoplasma DNA as a control. To test for mycoplasma, simply collect and lyse cells from your culture, perform a touchdown PCR, and run the product on a gel. If the cell culture is contaminated, a band will appear at the same location as a positive control. Cross-contamination of one cell type with another is also a problem. If the contamination is a particular fast-growing cell, the original cell line can be gradually replaced with the invading cells. As a result, an entire cell line in one's depository can be completely overwritten with a new cell. Furthermore, morphology is often not reliable enough to distinguish between the original and invading hosts, particularly if they're the same cell type, like one endothelial cell invading another endothelial cell. So there are a few common causes for this kind of contamination. Researchers can contaminate their own cell lines by working with multiple cell lines at a time. This often occurs when using the same pipette tips or aspirators with vessels containing different cell types. We recommend only working with one cell line at a time under the hood, if at all possible. More commonly, researchers receive cell lines from other labs or companies that were already cross-contaminated, so the importance of this cannot be understated. It has been estimated that 20% of scientific publications use misidentified cell cultures. This has also resulted in about 50% of preclinical research being non-reproducible. As a service, ATCC offers cell line authentication utilizing short tandem repeat profiling. STR profiling helps to detect misidentified, cross-contaminated, or genetically drifted cells, which invalidate research results. To take advantage of this service, simply receive a sample packet through the mail, spot cells onto a paper insert, and mail back. You will receive a report that includes the STR profile of your cell line, a comparison of your cells against an STR profile database, electropherogram supporting the allele cause at each loci, and a complete interpretation of the results. I also like to note that when you get back into the lab, now is a good time to start with freshly authenticated cells, and we do now offer human and mouse authenticated cells. So here I'm going to change focus and go into how you can choose the best model. And we're talking about which best cell model and why you would want to choose this. But before you go into that, I want to briefly discuss media before you go on to the models. So in general, continuous cell lines use a classical cell culture medium, such as DMM, RPMI, 1640, etc., usually with 10% FBS. More specialized cell types like primary cells, H-church cell lines, stem cells, tend to require more specialized media. It is generally recommended to maintain cells in the same medium, but if transferring to a new medium, it should be done gradually with the 50-50 mix of old and new, eventually reaching the new medium entirely. ATCC does not recommend using heat inactivated FBS under normal circumstances because it can denature growth factors necessary for the health of the cell. There are several types of cell models to choose from when designing your experiment, each with its own set of pros and cons. So let's start with the far right, the continuous cell lines. Continuous cell lines are often derived from a tumor. They're generally very easy to culture and have a low cost of culturing. They do not require a complex growth medium, usually just one of the classical cell culture formulations like DMM or RPMI supplemented with FBS. Like a tumor, they have great proliferative capacity. They all originate from a single clone, so there's minimal inter-experimental reproducibility. However, they are by nature abnormal cells with high instability and thus provide low in vivo predictability and limited biological relevance. The next model cell type that I would like to discuss is H-church immortalized cells. These cells are genetically modified primary cells such that the cell no longer undergoes senescence. So what it's created is a cell with continuous cell-like growth characteristics, but primary cell physiology. This means high proliferative capacity, and since every H-church cell type originates from a single transfected clone, there's no inter-experimental variability. Every lot will be virtually identical. Since the starting material is a normal primary cell, there is greater genetic stability with near normal primary physiology, so therefore high predictability and high biological relevance. The third cell type is an induced pluripotent stem cell derived primary cell, or iPSC derived primary cell. This cell type is derived from a somatic cell line that is dedifferentiated to an embryonic state and then differentiated to a specific primary cell lineage using a specific cocktail of growth factors. iPSC-derived primary cells are usually generated from a single clone, so there's likely little worries regarding the introduction of biological variability. The main problem with stem cell-derived cells is that they are differentiated in the immature primary cell types, so not the best for predictability or biological relevance. Lastly, we'll discuss the cell type that is the best model for in vivo predictability and biological relevance, the primary cell. A primary cell is usually isolated from normal adult tissue, so no genetic instability. However, for large-scale screening, there is a potential problem, not enough cells from a given donor. The use of new donors will introduce biological variability in your experiments. So each model has its own strengths and weaknesses. However, as you move from basic research to more clinical-like applications, you need to choose the model that most positively matches up to your research needs. As a final note regarding choice of cell model, continuous cell lines are derived again from a tumor or mutated in some fashion, such that the cell has deviated from its original source. So in every experiment involving continuous cell lines, the corresponding primary cell should be run as a control. That way, if you observe a specific effect on your experiment, you can be sure the results are valid and not an artifact of the tumor or mutation. So the take home is use primary cells as controls when at all possible. Okay, we're going to switch gears again and talk a little bit about transfections and what transfection reagents you can use. So now I'm going to discuss the basic method of lipid-based transfection. Since transfection is a method for introducing exogenous nucleic acid sequences into cells, there are various ways to accomplish this. One of the most common is lipid-mediated transfection. Lipid-mediated transfection is the most accessible and usually the simplest method available. It's based upon the idea that opposites attract. The transfection reagent is a cationic lipid that forms micelles with a net positive charge. Nucleic acids have a net negative charge, allowing the formation of complexes of lipid and DNA. The nucleic acid is covered by the micelle, so the complex now has a net positive charge on the outside. The surface of a cell has a net negative charge, so the complex moves to the surface and binds to the cell membrane. The nucleic acid lipid complex can now move through the cell's lipid membrane, allowing the nucleic acid to enter the cell cytoplasm. The nucleic acid then enters the nucleus, and the gene product can be expressed. ATC supplies two lipid reagents for transfection, GeneX Plus and Transfex. Transfex is our all-purpose reagent working well with continuous cell lines in primary cells. As suspension cells are a bit more challenging for transfection, we often find that GeneX Plus offers higher transfection efficiency for these suspension cells. So it's very easy to actually set up a lipid transfection process. And here I'm going to go through a simple workflow to demonstrate this. You always want to transfect cells when they're in the exponential growth phase. So you seed your cells to be ready the day before you plan to do your transfection experiments. On transfection day, which is day 0 on the slide, you combine your DNA with your lipid reagent and allow the complexes to form. Then you can add the complexes to your cells that you seeded on day -1. It's at the very top of the chart. You should then be able to detect protein expression at about 24 to 72 hours post-transfection. So now we're going to sort of talk about transfection best practices and how to optimize culture conditions. And these are 4 things to take into consideration. So even though it is a fairly straightforward protocol, there are several aspects of the process that you need to be optimized to ensure the highest potential, the possible transfection efficiency. To achieve a good transfection, the health of your cells is of utmost importance. The cells should preferably be at a low passage number. If they're seeded too high or too low of a density, the metabolic rate could be negatively affected, and cells could find the complex as toxic or may not readily express the gene product of interest. Contamination can lower the observed transfection efficiency, especially if it's mycoplasma. If you ever observe a drop in efficiency with the transfection protocol that has been previously optimized, mycoplasma contamination may be a likely reason. Media used during the transfection process is also a consideration. Certain lipids require no or low serum during the transfection process, so always follow the lipid manufacturer's guidelines. So a little bit more on transfection best practices. So for nucleic acids, which are DNA or RNA, one should use high purity and endotoxin-free material to achieve the highest transfection efficiency possible. Endotoxin appears to lower transfection efficiency. For DNA, one should verify promoter strength for the given cell being transfected. Certain promoters are known to work better with certain cell types. Plasma size is also a concern. The larger the plasmid, the lower the expected transfection efficiency. You should also verify that you are using a complete sequence in your plasmid. DNA plasmids that have been stored for some time can become degraded or nicked. So when designing your transfection experiment, you need to determine what assay methods you will employ at the timing for the data collection. As your protein of interest is expressed, you can use real-time PCR to detect RNA, probably 18 to 48 hours post-transfection. If this is your first time working with proteins, I would suggest running a time course analysis at a few different time points. If you plan to detect at the protein level, you need to give the cells enough time to make your protein and then perform any analysis before the protein is turned over. If you are not sure of your protein's half-life in the cell, again, a time course analysis is warranted. So of course, depending on the nature of your protein, there may be additional functional assays you can run. However, I would always recommend running analysis at the genomic and protein level, at least until optimal transfection parameters have been established. So regarding transfection reagents, there are 3 main areas of concern. First, which lipid should I use? That would depend on the cell type you're using and the cell characteristics. As I just mentioned, certain lipids are generally geared towards cell suspension, like ATCC's Gen X Plus. Other considerations involve what are you trying to deliver to the cells. Certain lipids are for DNA, others may be designed to work with RNA or small proteins. Next, you often have to optimize the ratio of lipid to DNA for the cell type and substrate that you're working with. Most manufacturers will provide guidelines, but some optimization should be expected. Lastly, one should optimize the incubation time for the lipid in contact with cells. If you're using a low or no serum transfection medium, you do not want to leave the cells in the medium for too long, and some complexes can become toxic to cells. So again, most suppliers will provide guidelines, so optimization may be required for your specific conditions. And so this is the very last slide on transfection, and it's going to summarize best practices on the transfection section of this talk. So to achieve optimal transfection efficiency, use healthy cells in the exponential growth, free of contamination, DNA that is of high purity and endotoxin free, and the recommended lipid for each cell type and substrate should be determined. Every setup may require some optimization, including but not limited to seating density of cells and the ratio of lipid to substrate. And so now we're going to move to the last major topic of this discussion, and this is going to be viability assays. So to cover growth and viability, we want to discuss ways of actually measuring the number of cells in a cell culture vessel and determine the proliferation or growth rate. This is ideal for assays in which you want to compare normal growing cells against an experimental factor that would positively or negatively affect the growth. So these two assays that we offer, MTT and the XCT cell proliferation kits, utilize tetraxyllium salts to deliver a quantitative analysis of cell numbers. So this slide is a diagram that shows how this works inside of a cell. So you can imagine the blue spheres are cells, and the gray inner sphere is going to be the nucleus. So with the MTT assay, the tetraxyllium salt is introduced into the cell. Naturally occurring oxidation reactions within the cell, which is indicative of normally growing cells, will reduce the tetraxyllium into formazine. So the cells are then lysed with detergents, releasing the formazine, giving off a purple color. The more purple the media, the more metabolically active and growing the cells will be. So this assay is good for measuring things like cytotoxicity and apoptosis. And the XCT reaction on the right is very similar, but the reaction takes place at the surface and it gives off an orange color as an indicator. From a more practical point or a point of comparison, so both assays are designed for 96-well plates. And here we have an overview of what these assays are going to look like. So for both, they're plated with, in this example, with a cytotoxic test compound. With the MTT reagent at the top, there's an incubation period to allow the reagent to enter the cell. The detergent is added, allowing for the compound to be released in the media. At this point, it can take a few more hours for the reaction to complete. With the XCT reaction, the XCT reagent and activation agent are added at the same time, so the reaction starts immediately. So one just practical benefit of the XCT kit over the MTT is there's one less step in the protocol, which will knock off a couple of hours from the whole process. And so this graph shows an example of absorbance versus cell number when comparing the MTT kit and the XCT kit. The XCT is in the blue at the top, the MTT in the red at the bottom. So in this case, eventually the absorbance will saturate and no longer indicate the number of cells as the number of cells increase in your plate. So the reading is taken on the growth curve. So this shows that with the XCT kit, it's more sensitive than the MTT, and that they can read cells between 5,000 to 30,000 cells. So with the MTT, the reading is between 5,000 and 15,000 cells. So the XCT assay, because of its fewer steps in the protocol and its increased sensitivity, is generally considered to be a more sophisticated assay. And so now we're reaching the end, and this is just one last summary on everything that we've talked about. So as a summary of what we've discussed, remember to use best practices to eliminate contamination while ensuring optimal growth and storage. In your cell culture workflow, choose the best model for your needs. Cell lines for standardization and primary cells as optimal controls that maintain biological relevance. H-turn immortalized cells can be the best of both worlds with continuous growth and primary cell characteristics. And so the key to good transfection practices rely on the use of healthy cells, purified substrates, and optimization of the lipid to substrate ratio. Lastly, you can monitor cell health and viability with the MTT and HTT viability assays. And that concludes this webinar on basic cell culture handling. And I will turn it over to Dr. Shapiro for further announcements.
All right, well, thank you, Steve. In just a few moments, we will begin our Q&A session. Please use the chat function available through the webinar program to submit your questions. The recorded webinar presentation will be available on demand on the ATCC website at www.atcc.org. Okay, let's start with our first question. Does the Mycoplasma detection kit quantify or identify the Mycoplasma contaminants?
Okay, and this is Steve. And just to be sure, can you hear my mic? Okay.
Oh yeah, loud and clear.
Okay, sure, thanks. So the question is, does it quantify or identify the Mycoplasma contaminant? It does neither. It's designed to be sensitive enough to read at the femtosecond levels of DNA. So it should indicate, any mycoplasma present, and only mycoplasma, and no other bacteria, microbe, or human cells will be read on this PCR assay. But it is not it is just an all or nothing assay. If the mycoplasma is positive, it just shows up on the band where the positive marker is on the positive control. So it doesn't quantify how much, and it doesn't identify the mycoplasma. Again, the idea is if you have determined that you do have mycoplasma contamination, the only real solution is to get rid of your sample. So the exact species really isn't that necessary.
Okay, good question. The next one is, can we use contaminated culture after treating with antibiotics? Good question.
Generally speaking, it's not recommended to use cells after they've been contaminated, or to try to treat them with antibiotics. It's difficult to really cure a cell culture that's been infected, and the amount of antibiotics you're going to use is probably going to be pretty toxic to the cells themselves. If it is for some reason, a very special cell line, and you really want to try, and it is possible, but as a general rule, it's very difficult to do, and it's never recommended. So if you have, same thing was with the mycoplasma. If you have contaminated samples or contaminated cell cultures, you need to simply discard them.
Okay, good answer. Now, for the transfection reagents offered by ATCC, do we have any optimized protocols?
So we do on both the Transfex and GeneX Plus. So we have a couple of different things on the Transfex and GeneX Plus product pages on our website. We have sort of general protocols for each, and you can use those and you can optimize them yourselves. But for the Transfex specifically, we do have a number of cell lines, primary cells, and stem cells that we have optimized the protocols for, and you could find those on the individual cell web page. I believe if you go to, this is under the document section on all of our website, on all of our product pages, there's a section for documents, and that's where we have this listed. But again, if you go to the Transfex web product page, there's a list of all the cell types that we've optimized Transfex for, and there's a link to that cell page where you can find those specific protocols. And for GeneX Plus, we have a couple. We don't have as many specific protocols, but we have sort of two general ones. One for the suspension cell lines and one for the adherent cell lines.
All right, all right. Wow, the questions keep rolling in. For cryopreservation, what kind of freezing container do you recommend for stem cells or primary cell? Good question. Too difficult to culture cells.
Yeah, so for these, there's a couple of different containers that you can use. One that we've tried with stem cells, primary cells, traditional cell lines, and even organoids is the polyethylene foam container that we offer, and you can get other places as well, the cool cell, which is, if you remember on the PowerPoint, there's the, it was that one of the cryopreservation pages, it was the green little container there, and they're very useful. And I don't know if you've used other containers, like the plastic containers that contain the alcohol that can get, when you put them in -70, they get very cold to the touch. The lids tend to stick, but the containers are, they work for all the different cell types. And this sort of foam material allows it to not be so cold when you take it out, so it's not too cold to the touch, and the lid comes off easily and things of that nature. So yeah, the cool cell that we offer has been worked with stem cells, primary cells, and organoids that we've tried in-house. And it's good because it freezes them down in 4 to 24 hours. So you can either freeze them in the morning, and in the afternoon take them and put them in the liquid nitrogen, or do it in the afternoon and just leave it overnight. So you have a little bit of flexibility there.
Great, great. All right, here's a really good question. It's affected me before in the past when I was in the lab. But cell detachment, right? What are some of the probable reasons for cells detaching from the plates unexpectedly?
A couple of different things. Probably the most common reason is due to the pH being not optimal, and so you're probably having incorrect buffering with your system. So this could be due to imbalance in the percent of CO2 in the incubator, or the amount of sodium bicarbonate you use in the media. But it's usually some sort of a pH problem that causes detachment. Also, another issue can be another real common one is simply, as stated in the presentation, if the confluency isn't correct. If the cells grow overly confluent, they start to sort of die and detach, as not enough room is available. And sometimes when you trypsinize your cells, if they're in the trypsin too long and you don't quench it appropriately, they may not attach correctly the next time around, and so they may just start to clump up and detach when you seed them the next time. So I would say, so those three things: the pH balance, the use of trypsin and the time you use of trypsin, and the confluency are three of the more common things.
Gotcha, gotcha. Okay, how many samples can be run with the mycoplasma detection kit?
If you follow the protocol as we suggest, you can run around 12 samples. There are a total of 40 reactions in the kit, but we recommend you run each sample 3 times, so you have twice as duplicates. One as a positive control with the sample, so you can test for various aspects of it. So if you run, it has 40 so different reactions, but you can run 12 different samples on that.
Alrighty, let's see. Oh, okay, so now we have a question about induced pluripotent stem cells and embryonic stem cells. So these cells grow in, they have a colony morphology. How should I count and seed them since they are unable to survive as single cells? Okay, so typically I'll go ahead and take this one actually. So what we recommend doing is, so first of all, when you dissociate them, use a stem cell rated dissociation reagent. Okay, ATCC has one. It's ACS 3020. And you do that at 37 degrees for between 5 and 8 minutes. So at this point, the edges are going to start curling off the plate. You gotta rinse off as much of that as possible. So you'll rinse the colonies twice with DMEM F12 to remove all the traces of this dissociation reagent. And then you'll want to scrape the colonies and pipette them, but be sure you do not over pipette them. Okay, just pipette them up and down 2 or 3 times. Okay, so and then once you remove the supernatant and resuspended the colonies, pipette them just 3 times. Don't over pipette them again, and dilute them to the appropriate ratio. And when you count them, just count the colony that you have. Don't try and count single cells. Okay.
Thanks for filtering that one, Brian.
Oh yeah, no problem, no problem. We've got another question actually that I can answer. Will the recording be available later? And the answer is yes. We're going to be, we have a long weekend coming up, so we're going to be targeting getting this on the web by next Wednesday. So it'll be available on www.atcc.org/webinars. Okay, got a question about heat inactivation. So why is heat inactivated FBS not recommended?
Yes, so heat inactivation, it basically denatures a lot of the enzymes and growth factors that's in the FBS that obviously helps cells grow. So in general, it's not recommended to heat inactivate it unless you have a specific need. Heating activation does inactivate the complement inside the FBS. So using certain assays that are immune related, you may want to use heat inactivation. But for general cell culture, it really doesn't really help. It just simply, like I said, you're denaturing a lot of the growth factors and things that are necessary for cell growth.
Okay, good answer. Now, this question's a little bit vague. Either the asker could get more specific. It says, what is the best passage for cell culture? And I guess you could answer this as when's the best time to passage the cells? What's the best confluence to passage the cells? Or how many passages should you use a cell before, just going to something fresh in your working cell bank? You still here, Steve?
Oh, I'm sorry. Sorry, I hit mute. My fault. Yeah. So yeah, general response to that is, again, if confluency is if you want to know how long you can grow them before passaging, again, 80% confluency is the general norm for most cells. As far as the number of passages, if the question is how many passages you can allow before the cell type is no longer good. For cell lines, again, they're pretty much indefinite. They can be passaged multiple times. There's no real limit on that. These are the classical cell lines that we offer on primary cells. Generally, you're looking at a maximum of roughly 10 to 15 doublings, which is 7 to 10 passages is generally the maximum, and it varies from different cell types. So always look at the manufacturer's or the company's recommendation. But for primary cells, you really want to you really want to keep those under just a few passages. You may want to grow them out, maybe double them a couple of times to sort of get them up and running. But yeah, passages should be limited there.
Okay.
I think I answered the question. Yeah.
You did. I'd like to throw a couple more things on top of that, though. So you'll want to make sure your cells are performing correctly right through all this. So say if the cells are no longer secreting a protein you're expecting them to secrete, or showing the morphology that they're supposed to show, you know, that might be an indicator that it's time to stop passaging and go to your working cell bank. That might mean that it's also time to authenticate your cells to make sure that they are the cells that you're supposed to be working with, right?
That's correct. Yeah. Good
So,
add-on to that.
yep. And so we generally recommend that every 10 passages or so, you should think about authenticating your cells, right? So, and that authentication could be via STR profiling, human STR profiling, it's a human cell line, mouse STR profiling if it's a mouse cell line, or we're looking for mycoplasma contamination as well. Okay, so let's see. Here's an interesting question about mycoplasma strains. Are there mycoplasma strains not detected by the universal mycoplasma kit?
That's a good question. So the kit detects, the vast majority, like 95% of mostly known mycoplasma, and that's about um, 60 or so species, I believe, is what we have. And then again, as I stated in the presentation, most mycoplasma species are the vast majority are a few common species, and those are picked up by the kit. So by those sheer numbers, the kit is going to be good for virtually any mycoplasma that is out there.
Right, but it hits the main adventitious
That's correct.
mycoplasmas. All right, good. Okay. Here's a couple questions around the cell proliferation assays that you talked about. The first one is, how does the XTT reagent recognize live cells versus dead cells?
So the way the both assays the way it sort of works from a pure scientific perspective. So as cells, the salts that are used, or if a cell is living, um, the salts that are used get oxidized normally, and this occurs at the surface of the cell membrane with the XCT kit. So if a cell is growing and living, the tetrazonium salts that we put in, they get oxidized, which causes that color change. And the more the color change is, the more the cells are alive. If the cells are dying or distressed, then you're not going to have as much oxidation, so then the color changes as much. So the basic principle behind the kit is it's an oxidation reaction. As long as the cell is living, it's going to oxidize those salts and change that color.
Okay. Question number 2 around proliferation assays. What is the best, MTT or XTT, and why?
Which is between those two, which would be the best one?
Yeah, and they say
Right.
for cytotoxicity assays,
Sure.
if that makes a difference in your answer.
Sure. Well, so I would say the biggest advantage of the XCT kit is, first of all, there are fewer steps involved in it, so it's going to be quicker by a couple of hours. And also, when you run both of them, it's more sensitive in that it will detect differences in lower cell counts. So there's this ideal range of cells that if it's too high, it becomes saturated and you can't really tell. Between 50,000 and 100,000 cells, you can't really tell the difference, but you can tell the difference between 5,000 and 10,000 cells. The XCT is more sensitive and you can detect lower cell numbers, so you have a more precise range. So it's two things: it's quicker and it's slightly more. It's more accurate at lower cell levels.
Okay, good answer. Here, the next one is, where do we find cell line authentication service on the websites? And you can go to if you go to our website, click on services, and that will send you to the cell authentication services. Also, there's the friendly URLs, www.atcc.org/str for human profiling, and for mouse STR profiling, it's www.atcc.org/mouse-str. And then the next question is, can I get the presentation on email if it's possible? So unfortunately, the presentation is going to be too large. However, we'll be sending an email out to everyone with the location of where you can watch the video on demand. All right. Steve, this is an interesting question that's coming next. That's about one of your other projects. And, you know, if we don't have a good answer, we can always send it over to R&D to answer later. But I am using the transfected 4T1 luciferase expressing cell line to obtain an in vivo model. Where can I get the protocol that you used to obtain the tumor model described in the product data sheet? Since each time I inoculated the cells, tumor initially increases in size, and then it's eliminated from the body of the animal. By the third week post-injection, I have no tumors.
Okay, so is the first question was about where can you get the protocol on? Yep. This is for the in vivo model, so injecting into the animal. We use, so the general protocol for the injection process is on the website on the individual page, and it refers back to the Perkin-Elmer protocol, because they're the ones that you use the instrumentation for the imaging. So they have protocols that we recommend for that. But we can certainly, I know, Brian, you can get that. We can certainly send that link to send that protocol to them. And as far as what you're seeing with the tumor growth, we can certainly get. I can contact tech support or talk to the R&D scientists, and we can talk about that. We can give an answer to that. So if you can forward this to me, we can return with some information.
Sure. Well, I'm happy to do that. So we will be getting back to you with some answers. Okay, let's see. Here's a question about trypsinization. And I assume that this is for trying to detach cells from the plate. So I use 0.05% trypsin, and it requires more than 5 minutes for trypsinization. I guess the question is would you recommend upping the dose of? Oh yeah, here's the part 2 of the question. Can I use a higher percentage of trypsin because they do not have an agitator incubator to use the lower concentration with?
Okay, so I would probably need to know what did they say what cell type this was?
Yeah, we don't have a cell type. If you're still listening, if you could send us what the cell type is, and maybe we can follow up with this question.
Sure. But yeah, the short answer, if it's a primary cell, it needs the lower trypsinization, or the lower trypsin concentration, I mean, and that does require a bit more agitation. And if it's a cell line, probably, or some other, other types of cells, you wouldn't need that low of a concentration. But definitely we can get back to you, and if nothing else, we can get some more detailed advice with tech support. But we can again, we can give you some more information on that. But we would need to know what cell type that is.
Okay. Yeah, I'm just rolling down to see if we have any. Oh, okay. Here we go. HEC293 Cas9 cells. So that would be the HEC293 cell line that expresses the Cas9 enzyme. Mm-hmm. So a recent release.
Oh, I'm sorry. Yeah.
Thank you for buying that.
Right. So that's the cell type that he was referring to about the trypsinization.
Yeah. Okay. That's weird because HEC cells, in my experience, they dissociate pretty easily.
Mm-hmm. Yeah. So we'll look into that. That's a special type of cell that there are other people in in ATCC that are experts on that. And I haven't been working on that project, but that's definitely we can definitely get back to you on that. That's a good question though.
Okay, okay, good, good. All right, let me see. Um, okay, how do we authenticate our current cell lines? Um, that I guess would all depend on if the cell lines are human in origin, animal in origin, or of an altogether different species, right? So there's human STR for human cell lines, mouse STR for mouse cell lines, and then if you want to determine if the species of a different cell line, you could do a CO1 barcoding, right?
Yes, correct.
That's cytochrome 1 oxidase barcoding, which gives you, we can do that test for you, and we can identify 14 different species of animal via that. And of course, you can also always test for mycoplasma contamination, which is another method of authentication. All right, so let's roll to the next question. What cell lines have been evaluated for transfection with GenX Plus?
With GenX Plus, we have again we have two general protocols for that. One for general suspension cells and one for adherent cells. We don't have as many optimized protocols as we do for transfecs. But if you go to the GenX Plus product page, the protocol will be there, and if there are any specific ones, it will be listed there. But most of our optimization has been done for transfecs.
Okay. Is there a good handbook or book useful for cell culture? More details. Steve, this is a great time to plug our cell culture manuals, or
Right.
our cell culture guide rather, and you can plug the latest addition to those.
Sure. So on our website, we have various cell culture guides for cell lines, primary cells, HTERT, iPSC stem cells, so specific ones for all of those. In addition, a lot of the microbial culture guides, and we've just come out with the more recent one for the organoids that we have with the HCMI models, specifically for the organoids of the HCMI models, and it will be added on to later as we add some more products to it that will sort of help with that. But it is a good general cell culture guide for the organoids as well. So we have guides available on our website for primary cells, cell lines, stem cells, HTERT mortalized cells, and the organoid models.
Right, and these guides contain all the information that you need to cryopreserve your cells, to passage your cells, thaw your cells, as well as there's some details about cell counting and that sort of thing. Well, so really good, solid basis for helping you culture your cells. All right, let me see. Any suggestions for a good neural cell line? So I could kind of speak to this a little bit. I mean, it sort of depends if you're looking for just something that's easy to passage and grow. You know, there's the sushi 5Y cells that you can use. There's also some neuroadrenal cell lines that are out there. If you want something that you can differentiate down one of the three major neural lineages, though, I'd recommend the neural progenitor cells that we have. So these neural progenitor cells, you know, you can differentiate into neurons, astrocytes, or oligodendrocytes. We also have media supplements that you can use to make that happen, as well as just growth media for these guys. And actually, I've done a couple of webinars on the neural progenitor cells. So if you look back through some of our old webinars, you'll find them. So yeah, neural progenitor cells from ATCC. You can also find them on our website, www.atcc.org/neuro. You can find them there.
And also on that, we also have, and this, if this is something that you can use, we also have some glioblastoma models, cancer models that are neurospheres. So they're like 3D models. They're part of the HCMI program, and they're not exactly organoids, but again, they're neurospheres. So they're like these 3D spheroids of glioblastoma models. We have several of those available.
Oh yeah, that's right. That's right. I should have known that. Okay, so wow, this is like the longest Q&A session ever, but let's keep rolling. Do you recommend titrating cells after cell detachment prior to replating? And I think a better way of asking this question is, do you recommend counting cells after detaching them before you replate them?
After detachment, before replating them, you can. I think generally when you freeze them down, you always want to count when you thaw. That's really the more important thing. So I mean that certainly is an option, but again, after you thaw, counting is really you'd want to do that there. But do I recommend it? I mean, maybe not, because again, time is kind of the essence. If you're detaching them, you want to kind of freeze them down sort of relatively quickly.
Okay. All right. Let's see. Here's a question that we may not be able to answer. We can certainly refer it over to our technical service representatives, but do you have any recommendations for validating any human fibroblast primary cell culture? I'm not really sure what they mean by validation. I mean, is it validation of function, or just that they are what they are?
If it's just validating to make sure they are fibroblasts, the best thing is to use of markers that you can look on our website and see the markers that we use. The ones that are positive and negative for fibroblasts. But I guess surface markers, through immunohistochemistry would do that.
Right. I do recall that for most of the human primary fibroblasts we do staining for, is it TE7 is the that fibroblast marker that we use?
Oh, sorry. Yes.
Okay. All right, and then we do negative staining. I mean, we stain for also von Willebrand factor. That would be, a negative marker, so we don't expect to see any staining for that, and also cytokeratin staining. All right, let me see. Okay, here's another question. How long can you leave the frozen aliquots in -70 before transferring to -40? Basically, is it not a good practice to leave them in -70 for an extended period of time, is the follow-up?
Minus aliquots of reagents.
Yeah.
Well, whatever the reagent is, they all have their recommended storage temperature. So it depends on what that is. If you're speaking of cells going from -70 to -140, if that's what they're talking about, the cells, yeah, they generally go in the -70 for 4 hours to 24 hours, depending upon what you're using to freeze them down, and then they go in the -140. But if you're talking about reagents, it depends on whatever the reagent is. to be honest.
Mhm. I think what they mean is, I don't know, I've done this sort of thing, in the past, I hate to admit, but where you put the aliquots of cells that you've frozen down in the -70, and then you went off and you did something and you just kind of lost track of them and left them in the -70. Um, and then maybe, a couple days later you slap yourself on the forehead and say, "Oh, I meant to put them in the -40." You know, how long are they still good in -70? Is it,
Right. Yeah. I think for most reagents, and again, that again, it depends on the individual reagent, but it's anything that's colder is always better, or is always fine. But the thing about reagents, it has more to do with if it's in a temperature that's too cold for too long, that could affect the container that it's in, the bottle or whatever it's in, because certain containers aren't designed for really cold temperatures. So that really is what you have to look into,
Mhm.
and because that could cause the containers to crack. But in general, if the reagent is okay at -40, being
Uh-huh.
colder
Hey,
doesn't really. Yep.
Steve, we just got an update. She means specifically, or he means specifically, aliquots of cells. So how long
Oh,
can you
okay.
leave your frozen down cells in -70 before moving them to -140?
Okay, sure. Sorry. Yeah, and that's I previously answered that. So yeah, for most containers, again, it depends on the container that you're using, and you should use a container that is designed specifically for freezing down cells. Like a lot of people will use just styrofoam containers. Those don't work really well. But like in most situations, it's about minimum of about 4 hours, and they can generally stay in there 24 hours for most devices. And so I would say 4 hours to 24 hours is the length of time it takes. But again, the container needs to be designed so that you have that cooling rate of around -1 degree Celsius or so. And if it's designed for cell freezing, then it should achieve that. And again, the example I gave earlier with the cool cell that we have, yeah, it takes about 4 hours to freeze them down, but again, they can stay for 24 hours without a problem as well.
Okay. Now I got another question just popped up that's related to this, so I'm gonna keep throwing this at you. Can we store cancer cell lines in -80 for a long time, like 6 to 8 months, instead of liquid nitrogen, or will this negatively affect the cells?
For yes, humans for cell lines for 6 to 8 months, that's a long time for that temperature. So I would say no. I mean, yeah, I'm not going to say what's possible and what's impossible, but that would definitely have a negative impact on the cells.
Right. I gotta go along with that. I mean, you want
Yeah.
to have the cells at, the -40 degrees because you want like all metabolic activity to stop, you know. And even at -70, something could be going on. It might be going very slowly. It might not be very likely that's going to happen, but that the chance is there that you'll have see some changes.
That's
Okay.
correct. That's correct.
All right, cool. I think we talked about that as much as we can. Okay, we got another one here regarding the transfection. Why in some publications the same transfection reagent is used for siRNA delivery and into cytoplasm and crRNA's delivery into the nucleus? What controls the delivery of nucleic acids when using lipids to cytoplasm or nucleus? Is it the volume of the transfection reagent? And maybe that might be a better way of putting that. Is it the ratio of transfection reagents to nucleic acid?
I think if I got that question correctly, as far as what determines whether it goes into the nucleus or the cytoplasm.
Hmm.
Yeah, so it's the optimization of a lot of different things. It's the optimization of the ratio of the nucleic acid and the lipid compound. Um, I mean, a lot of, and that will, the better the optimization will determine whether it goes into the nucleus the best or not. I mean, it's probably not going to be 100% either way. Well, it's definitely going to be 100% either way. It's a little bit random when doing a transfection reaction. It goes to the cytoplasm, and it can either stay in the cytoplasm or transfect into the nucleus. You can't directly control that other than by optimizing that ratio. You increase your chances of getting it into the nucleus.
Okay. All right. Let's see. Um, oh, okay. So there's the follow-up, by the way, to the questions we were talking about the -80 questions were from some folks that don't have liquid nitrogen in their laboratory. Um, so I guess we don't have really much of a good answer for that. Um, ah, do we have a protocol for checking expression of Cas9 when people are using the new HEC293 Cas9 expressing cells?
Yeah, that's a question we can probably have to get back to you on that. So yeah, the Cas9 cells, they're specific cell line and yeah, or a very specific cell type. So yeah, we can get back to you on that question.
Yeah, I would agree. I mean, I feel like we check expression by Western blot, but I am not entirely sure of that
Oh, sure,
myself.
I'm sure. Yeah, there are conventional ways of doing it. Yeah, you're right, with things like that, Western blots and so forth. But as far as the best way and what to expect, yeah, we can get more information on that.
Hmm hmm all right, let's see. Um, here's a question that will probably be sent over to our TSRs, but I wish to do primary culture from fish tissues. How do you facilitate easy attachment of tissue during primary culture using tissue exponentiation method? I guess, how do you facilitate easy attachment um, of cells during primary culture without using serum?
I'm not sure I got that question.
Okay, so I actually kind of have an idea of what they might be talking about here because I used to do, actually as a grad student, I did almost all primary culture and very little in the way of culturing like classical cells unless they were workhorses for some other project that I had. So, for primary culture, you want to avoid using serum in your media, right? Um, and
Correct.
there's two ways of doing this. One way would be using a little bit of serum when you initiate the culture and then remove the serum and replacement with something like BPE, which would be bovine pituitary extract. So that's one way that you could do it. The other way that you could do it is just do it with just totally eliminate that the serum and just use the pituitary extract, the bovine pituitary extract. So that's just one way that I would suggest doing it. Again, you can contact our TSRs, our technical service reps, for more information about that. All right.
Yeah, that's great. I would advise that.
Yeah. Yeah. Okay. Let's see. Interesting. Okay, I'm wondering if the COVID-19 infected staph culture cells at facility. Is it possible for COVID-19 to infect cell cultures just in general? Is it possible? And then there's something about how to avoid at the BSL-2 level. So, I mean, I kind of feel like generally speaking, COVID-19 can infect anything with two receptors, right? The ACE2 receptor and the other TMP receptor. I forget exactly what the name of it is, right? So, so it would be possible to um, if you are, wearing PPE like you should in the laboratory and performing your work under, sterile conditions like you're supposed to, that should not be a problem. And then the second question is how to avoid a BSL-2 level. There are some BSL, if you say you're doing research, like with COVID-19, there are inactivated materials that you can use, and there's, I believe, synthetic standards that you can use that ATCC provides. And those you can find at www.atcc.org/coronavirus. All right, let's see, what else do we have? I mean, the facility is a BSL-2 level. Okay. So back to that previous question. Yeah, BSL-2 facility, that should be PPE as well as using being sure it's in a biosafety cabinet, laminar flow, and all of that. And so you should as long as you're cleaning everything like you should in between uses in the hood. But the staff should, which kind of PPE? That would be gloves, gown, goggles, and yeah. So gown covering your arms, fresh gloves every time, and goggles.
And also, I always remember with gloves, you always want those to go over the sleeves so that there's no space between the sleeves and the gloves, if that makes sense, so that it's sort of tugged in a little bit.
Right. Well, at ATCC, we regularly use those in our BSL-2 facility, right? But that is pretty much just what we do. That's just part of the processes that we have in place. Okay, well, I think that we've caught all the questions that have come in. Any others can be sent to the technical service representatives at ATCC. And also, we have more webinars that are coming in soon. In a couple of weeks, on June 4th, I'll be presenting my talk on iPSC-derived primary cells and how they can expand cell-based assays with an unlimited biologically relevant source. On June 14th, Brittany Tang will be providing an educational webinar on COVID-19, highlighting ATCC's tools to fight the pandemic. And then on June 25th, Dr. Tasha Santiago Rodriguez will give her talk on viral metagenomics and the use of standards from biology to clinical applications. So thank you again, everybody, for an especially good Q&A session, and have a great day.
Thank you, Brian.
Thanks all.